The authors of this paper aim to investigate how dynamic RAS-ERK signal inputs are interpreted at the level of gene expression, and whether altering this RAS-ERK signal transmission with the drug SB590885 disrupts this interpretation. So this RAS-ERK is a part of the MAPK signaling network and is frequently dysregulated in cancers. So how this pathway works, as illustrated in this middle part here, is that growth factors will bind to the receptor of kinases on the cell surface. This leads to the activation of this RAS. It's a small GPTase that acts as a molecular switch. This then activates RAF, another kinase. Then this RAF phosphorylates this MEK, which then phosphorylates this ERK. And this ERK is the key effector here and will then enter the nucleus to regulate gene expression. So kind of the hypothesis illustrated in this figure, part of this figure, is that cells use dynamic patterns of RAS-ERK signaling to regulate this gene expression. But if this pathway is altered, in this case with this SB drug, the cells might misinterpret this input pulse signal and lead to atypical gene expression. So for the methods used here, one is being optogenetic stimulation. Cells were exposed to two different patterns of RAS activation. There's this constant activation and then this pulsed stimulation. So this is just more of an intermittent RAS activation that can mimic natural signaling fluctuations. This is more of a control to go back to. The cell types used in this were wild type NIH3T3 fibroblasts. As seen up here, has normal kinetics. This down here is the same cell type, but with this SB drug. This is a RAF inhibitor known to prolong this RAS-ERK pathway, just delays it a bit compared to the wild type. And then over here we have our gene expression readout. So after the stimulation of constant and pulsed, the cells were fixed and stained for three key RAS-ERK target genes. One being the c-Jun up here, a transcription factor that is linked to cell proliferation. And then this ERG1, again, is a gene that adapts to sustained ERK activation. And then lastly, the cyclin D1 is a regulatory factor in the S cell cycle transition, again, promoting this idea of proliferation that we would see a difference in with this constant or pulsed and with this RAF off delay drug. This section gives a more quantitative output of how the gene expression is affected. We have the two cell types over here. The purple is the fibroblast with the drug applied under a pulse stimulation. And then the fibroblast with no drug is shown with the two lines. The dotted line is constant stimulation, and then the pulsed is the solid line. The pulse stimulation was done 30 minutes on, 30 minutes off, again to test how cells filter the dynamic signals. And then the continuous activation acts as a reference to compare the sustained versus dynamic input responses. So to get these graphs, the protein expression was measured using immunofluorescence imaging with high content microscopy. So to do this, the authors fix the cells with PFA to preserve their state, and then permeabilize with detergents to allow antibodies to enter the cells. The immunofluorescence staining was done using primary antibodies specific to each of these three factors we wanted to look at. Secondary antibody was then added so that they could actually look at it under a microscope. And then the cells were imaged using a high content fluorescence microscope that allowed for very precise single cell analysis of the fluorescence intensity. Looking at all the cells, a median fluorescence intensity was found, looking in the nucleus for the transcription factors or the cytoplasm for the other proteins. and was quantified for thousands of cells, and they got those graphs. So looking at this more closely, there are some key findings. Looking at the normal cells, so the lines, we see that the c-Jun and the cyclin D1 were only strongly expressed under continuous stimulation, so that dotted line is a bit higher and kind of represented with that darker purple arrow, but not under the pulsed inputs. The EGR1 adapted to continuous stimulation, which you can see with that darker purple arrow pointing down, while the pulse stimulation maintained moderate expression. It didn't really change too much compared to the constant. Now looking at the SB drug treated cells, the pulse stimulation caused the c-Jun and the cyclin D1 to behave like they were continuously being stimulated, which indicates that there is a loss of dynamic filtering. The EGR1 expression in response to the pulse inputs now resembled its response to continuous activation, which is showing reduced adaptation. So overall, normally cells distinguish between transient and sustained signals, regulating gene expression accordingly. So when the RAS-ERK kinetics are extended, this distinction is lost. So the pulse inputs falsely trigger sustained gene expression, which could potentially lead to atypical cell behavior. So the authors then look specifically at what was happening to cell proliferation, trying to get a more quantified idea of what is going on. So kind of asking the question, does extending this RAS-ERK pathway activation with this SB drug increase the likelihood of cells entering the cell cycle? So the authors tested how different dynamic patterns of RAS activation, the continuous versus pulsed influence S phase entry or DNA synthesis, measured using EDU incorporation, as seen over here. So how this was done is after 19 hours of stimulation, cells were incubated with EDU. EDU is used to measure DNA synthesis and cell proliferation. Basically how it works is that during the S phase of the cell cycle, cells will incorporate thymidine into newly synthesized DNA. This EDU is structurally similar to thymidine, so it gets incorporated into replicating DNA instead of that natural thymidine. So high content fluorescence microscopy can then be used to measure the percentage of this EDU in each condition, which is being represented here in this part of the figure. But over here in the heat map, it visualizes the percentage of proliferating cells under different stimulation conditions. So this x-axis is the on interval, and then the y-axis is the off interval. And the color scale here is the higher the proliferation, or the more EDU in the cells, is shown in the brighter regions. So findings for the normal no drug cells, only long continuous RAS-ERK activation drove proliferation. Short pulses or the intermittent stimulation failed to induce significant cell cycle entry, meaning normal cells properly filtered out these weak signals. However, with the slowdown treated drugs with the SB drug, the proliferation response expanded to a broader range of input conditions. The cells entered the cycle in response to the pulse signals that normally wouldn't trigger proliferation, and this suggests that the prolonged ERK activation causes cells to misinterpret weaker signals or inputs as strong proliferative signals. So the last part of this figure just shows the experimental results in a conceptual model. Here, the normal cells are say that short-lived or low-intensity signals are filtered out, preventing any inappropriate responses. Only sustained, strong inputs lead to gene expression changes and proliferation. However, with the cells with altered RAS-ERK kinetics, the signal decay is slow that can lead to prolonged ERK activity even after input removal. The weak signals that should be ignored are now misinterpreted as strong signals, and this can lead to some inappropriate responses. And then going back to the fact that disruptions in this RAS-ERK signaling can lead to cancers, we can think of this as a way to show cancer mutations or certain targeted drugs may create a blurred perception of signals driving hyperproliferation where there shouldn't be. So with this figure, there's some strengths and weaknesses. The figure combines live cell optogenics, immunofluorescence imaging, and cell proliferation assays, so it takes in a lot of different data points to get to these conclusions. It also uses high content immunofluorescence and proliferation assays that allow for single cell resolution and large sample sizes. And this minimizes variability and can increase statistical robustness. There are some weaknesses, however. The study primarily only uses NIH3T3 fibroblasts rather than actual cancer cell lines. So, it's not very specific to in vivo models. The proliferation assays also only measure S-phate entry over 19 hours. Kind of begs the question, does this lead to actual sustained proliferation over multiple days, or why is it only 19 hours? And then lastly, it only examines that c-Jun, EGR1, and cyclin D1. While these are well-known RAS-ERK targets, they could continue this study and do a broader range of factors, maybe using RNA sequencing or different analyses tool to get a more well-rounded look at the RAS-ERK pathway implications.